Good evening, everybody. Happy Friday, celebration of microbial diversity. "50 years of microbial diversity," which means you're just getting started as a scientific community. Congratulations. I don't know if my boss is still here. He would appreciate that one. But my name is Sebastian Lay. I'm MBL's Chief Advancement Officer, and that means pretty much nothing tonight except to say these lectures are supported by philanthropy. They are being recorded. They are being simulcast on Zoom, and some of our fellow audience attendees will be able to ask questions during the Q&A, which will follow tonight's lecture. I ask you to please silence your cell phones because I know we're in for a great talk tonight. So please just double check that. There's nothing worse than in the middle some bizarre ring going off and we'll have to march you out into Yale Pond. And then finally tonight, penultimately, there is a reception following tonight's lecture on the back patio of Swope. So if you don't get an opportunity to interact with tonight's speaker by asking a question after the talk, you can join us for wine and beer or socialization afterwards under the tent, and I hope you will. This is the beginning of the celebration of the 50th anniversary of the course, and I think you'll hear a little bit more about that. And finally, it's my pleasure to introduce a friend and colleague, Colleen Cavanaugh, tonight, who will introduce tonight's lecturer. She is a very long-time member of the MBL community, a former trustee, a president of the MBL Society, which is the inheritor of the MBL Corporation, as many of you might know, a advisor, supporter, and contributor to this intellectual community who made me promise I would not say how many years she has been lecturing in the microbial diversity course, but I can promise you that it is a very significant amount of time that has had a serious impact on the field and this community. So, Colleen.
I've whited out the year of the picture of me in the course, so you can't figure it out. Thank you, Sebastian. I am very honored to introduce Dr. Carrie Harwood, the Gerald and Lynn Grinstein Endowed Professor in Microbiology at the University of Washington. Carrie is a renowned, world-renowned microbiologist with an amazing breadth and depth in research and scholarship. I have always admired her for her integration of bacterial physiology, molecular biology, environmental aspects, and evolution. And that she's taking a holistic view, even though, as you'll see, she tends to work on a single organism, but it's an ecosystem unto itself. Carrie was actually born in Boston and educated in New England, Colby, bachelor's, BU master's, I should know these by now, UMass Amherst, PhD, and then Yale for her postdoc. She has a long history at the MBL, both with the course, and has also served as a trustee. Among her many awards and honors, she's an elected member of the National Academy of Sciences, the American Academy of Arts and Sciences, and the American Academy of Microbiology. So Carrie's not only tonight's Friday evening lecture, but she's also the kickoff speaker for the 50th anniversary of the Microbial Diversity course, which starts tomorrow with an all-day symposium. I think she may talk about it more, but 8 a.m., be there, here, I think. Here? Here. So, wait a minute. I didn't want that. What happened? Who invited Ed? Could someone go back? This is it. There we go. Her title refers not only to the diversity course, 50 years, but if you change it just by a few years, it gives you the 3 billion years of microbial diversity and their evolution since they first were found. So a quick side trip. We live in a microbial world. For those of you who know the multitudes that comprise our own microbiome, which rival our own cell numbers, to the microbes in and on everything we encounter in daily life. And this is going to be a quick sideline. The term pathogen is okay to, yes, it causes disease, but let me point out, there is no such thing as a bad bacterium. There isn't. Get a grip. They just want to eat. You happen to be their food, perhaps, or plants, whatever. And they have evolved such exquisite ways of getting you to spill your guts, spill your cells, get between your cells. So, I mean, it's truly an evolutionary marvel. And as Gary Borsi put it tonight, it's not personal. So, but the sheer numbers that we are engaged with on a daily basis, typically in water, seawater, fresh water, tap water, bottled water, there are 10 to the 6 per mil, 1 million per milliliter, which gives you 5 million bacteria in a teaspoon of water. 500 mils in this bottle, minus a few tens of millions. And there are typically 1 billion bacteria per gram of sediment and soil. So it's a huge number, and the diversity is shocking through their billions of years of evolution in concert with the changing geochemistry of the Earth, and they have a plethora of metabolic capabilities that Carrie will be describing within a single bacterium, many of them. But they have the ability for photosynthesis, chemosynthesis, the ability to use iron or sulfur as energy sources, and can fix nitrogen into fertilizer, ammonium. While many of these capabilities are harnessed by humans, for us, for agriculture, medicine, industrial applications, most of these bacteria cannot be cultured, and therefore the diversity and the metabolic capabilities, the spectrum of metabolisms has not been known. With the advent and development of molecular sequencing and analysis, this diversity is being revealed, and we are truly in the midst of a microbial revolution. This is such a great time to be alive, as I am sure you will all admit. Seriously. So this revolution captivated Ed. Please change the slide. E.O. Wilson. And it literally did. I mean, and he stated in the final page of his autobiography, Naturalist, "If I could do it all over again, I would be a microbial ecologist. 10 billion bacteria live in a gram of ordinary soil, a mere pinch between thumb and forefinger. They represent thousands of species, almost none of which are known to science. Into that world, I would go with the aid of modern microscopy and molecular analysis to discover new life ways and alien food webs." And I have to say, Ed, I'm so thrilled that he did come to this realization as kind of the father of biodiversity, but Carrie was way ahead of E.O. Wilson. Early in her career, graduate career next, she actually took the microbial diversity course. You can see her there. And this is the course that was designed and run by Holger Janisch, which is in the upper right corner. But he conducted it at MBL and is one of the discovery courses that has been running for 50 years. Carrie has carried this love of microbial diversity through her entire career and to future generations as a co-director of the microbial diversity course from 2000, next slide please, 2000 to 2003 with Alfred Sporman. You can find Carrie, she's the only one with sunglasses, and Alfred is up at the top. Alfred was very sorry he couldn't be here tonight, and he noted that it was terrific being a co-director with Carrie, that they shared responsibilities, humor, and excitement about the same discoveries, which I would love to hear about. What happened back then? One of Carrie's many strengths is this integration of different mindsets from physiology to molecular to the whole organism. And this is taking a world view of bacteria rather than focusing on a single metabolic pathway or a single operon. And this has resulted in rapid advancement throughout her career with many discoveries. For example, she's known for her early and continuing studies on anaerobic biodegradation. For example, showing that soil bacteria could actually catabolize some of the most recalcitrant compounds, such as lignin and the lignin components, and chemicals such as benzene. These have obvious applications to carbon cycling, biofuels, and pollutant remediation. Recently, she and her group reported a new biological way, enzymatic synthesis of methane. Now, methane has always been thought to only be relegated to the methanogens, and now it was discovered in a bacteria, in a member of the bacteria rather than the archaea. And this again is her organism, discovered as one of the many metabolic talents of Rhodosudomonas palustris, our pal, alpha proteobacterium, and the model organism studied in her lab. This bacterium seemingly does it all. From photosynthesis to anaerobic biodegradation, nitrogen fixation can produce prodigious amounts of hydrogen. This may be the microbial fuel cell that keeps your car going soon. And there are new metabolic capabilities waiting to be discovered. Carrie will discuss this remarkable microbe in her talk. She refers to it as microbial diversity personified, and with its many talents and applications. So it is a pleasure to welcome Dr. Carrie Harwood back to MBL to celebrate microbial diversity and to learn about your research on the remarkable bacterial metabolic diversity, even within a single organism, our pal, which I actually think of as the Swiss Army knife of bacteria. Carrie.
Colleen is wearing our pal red.
Oh, that's right.
She's wearing our pal red.
The color of the cultures in one of the many modes of metabolism.
Thank you so much, everyone. It's a real honor to be giving this lecture on behalf of 50 years of microbial diversity, and so exciting to reconnect and see so many old friends here. I'm really very complimented. So we're here to celebrate the 50th-ish anniversary of the MBL Microbial Diversity course. And so today I want to say a little bit about the history, tonight about the history of the Microbial Diversity course, about how the course empowered me in my research program, and tell you a bit about my own research. So what is microbial diversity? Colleen gave a great description of microbial diversity. I have a slightly different description that I've arrived at that I use for myself. And to me, microbial diversity is things microbes can do that humans can't, with climate change being a notable exception. Some of these things are really important, like nitrogen fixation, biodegradation, host nutrition, infectious disease, antibiotic production, and some of these things were interesting, took the interest of students in this course, for example, and these interesting, but who knows why you would be interested in them, turn out to be important. For example, bioluminescence, which Woody Hastings studied, turned out to be the first example, and a simple example, that led to the discovery of quorum sensing, which is communication between bacteria. Biofilms are another, and radical SAM enzymes even are very important discoveries that have come out of studies of microbial diversity. Now, at the MBL, only part of the action in microbial diversity is in the lab. And I always thought that you couldn't have a really great microbial diversity course anyplace other than the physical place of Woods Hole, because it has so many great microbial habitats. And that's a large part of what makes the microbial diversity course so effective and fun. And so I want to take you on a little tour of Woods Hole from the microbial diversity perspective. And so we start here at something that's now called the Waterfront Park, but when I was a student, we called it Garbage Beach. Is it still called that? Yes, okay, all right, so good. So if you go to Garbage Beach, especially this time of year when the water's a little warmer, and you streak out a plate of seawater complete agar, always some of the colonies will be bioluminescent bacteria, well, a big plate of which is shown here. And this, I mean, I couldn't believe it. I couldn't believe that you could just streak seawater on a plate and get bioluminescent bacteria. It was really a thrill for me when I was a student. Then we go down from Garbage Beach along Bar Neck Road, and then we get to the Woods Hole Yacht Club, and right along the road in front of the yacht club are beach peas. And beach peas are legumes, which means that they have symbiotic bacteria in their roots, and they form nodules, these special structures that do nitrogen fixation, and so take nitrogen gas out of the air and provide nitrogen to the plant. And then come back to the lab, squish the nodules, and you get these characteristic mucoid colonies, which are some sort of rhizobium species. I bet someone in the course has figured out the exact species name for these bacteria, which I could not find looking in the literature. Then we go out Oyster Pond Road to Oyster Pond, which is brackish, but there's a place where the water flows in and out of the pond, seawater. And so it's sulfur compounds in the seawater mixing with oxygen. And so it's a great place for natural enrichments of sulfur oxidizing bacteria, which includes Beggiatoa, which are shown here. They convert hydrogen sulfide to sulfur, which is in granules. And actually, this photo was taken by a student in the course, Ralph Schauder. Then no placement icon is needed for the Sippowisset marshes. The Sippowisset marsh has microbial mats that are probably more extensive and structured than any other place in the world. I mean, you can call me on that if you want, but I don't know of any other place. And if you walk along the mat at low tide, it's very, very, it's sort of like walking on a putting green, but firmer, because you're walking across a mat of filaments of cyanobacteria. And if you dig into the mat, there's a whole organization of bacteria, starting with cyanobacteria, which make light, energy from light and are green, to other kinds of phototropic bacteria, like the purple sulfur bacteria, shown here, green sulfur. And these absorb light in the wavelengths that the cyanobacteria can't absorb. And they feed off of sulfide that bubbles up from below through the activity of sulfate-reducing bacteria. Now, at the beginning of the course, maybe the first night of the course, rain or shine, it doesn't matter if it's pouring out or not, you have the team building exercise, which is the cedar swamp and the Volta experiment. And this is like totally fun. Like, so you get so in the cedar swamp, which is like lots of dead leaves and stuff that you can stomp on when you go into the swamp, kind of up to here or here, depending how much rain there's been. Then methane comes bubbling up from below. And the idea is to capture it in a great big funnel that's stoppered at the top. So you get the funnel all full of methane, then you unstopper it, put a match in, and this is what you get. And it's like, you also, the novices usually get some singed eyebrows as well. So when I was involved in the course, we didn't have tweeting, but now we have tweeting. And so, and the pink berries, like, I know every single person that's taken the course knows about the pink berries, which are in Little Sippowisset Marsh. They're like a consortium of bacteria we know now. And they're just a really cool example of a couple different microbes living together and having sort of a cycle of food, passing food around. Then we take everything back to the lab. And this is Grace, who was in the course when I was director, looking at stuff under the microscope. It's important to have lots of junk around you, because that makes it more homey. And then we have the light table here, because phototrophs were, I always loved the phototrophs. And this is Rachel Whitaker pondering her phototrophs. She's current co-director of the course. And then these are our rewards, is we try, a big part of the course is enrichment and isolation, and we try to get the bacteria into captivity. Here's one that I really like, because this is a white colony with purple phototrophs growing around it. And they're doing some sort of nutrient exchange, which I don't know what it is, but maybe someone's figured it out in the interim. So this is an important part of the course, and I'm going to talk about other important part of the course, but I want to first talk about the course founder and first director, who was Holger Janisch. And Holger was a microbiologist, a scientist at Hui, and the world's greatest storyteller. And the hydrothermal vents hadn't been discovered when he started the course up, but he was an expert in bacterial sulfur metabolism. So he really appreciated and discovered that hydrothermal vents supported chemosynthetic bacteria as primary producers for deep sea ecosystems. And this is something that Colleen has worked on for her over the course of her career. And so Holger, it was sort of a surprise that the MBL asked Holger to develop a course, because the MBL was known for things on, big animals and sort of model systems, the giant squid axon and so forth. But they asked him to do it, and he modeled the course after a course established in 1930 at the Hopkins Marine Station in Monterey, California, by the Dutch microbiologist C.B. van Niel. And Van Niel course, the way Van Niel kind of brought microbial diversity to the United States in the 30s. And the Van Niel course was designed to bring microbes into captivity and to study their diverse physiologies and biochemistries. And many of the early instructors in the MBL microbial diversity course were alumni of the Van Niel course. So this was in 1971, but 1971 was not an auspicious time to start a microbial diversity course, because we were just in the middle of figuring out, it was a real burst of activity in molecular biology, and the central dogma of molecular biology reigns supreme, which of course is DNA makes RNA, makes protein, and all the different codes that have to be solved for living cells to do this. And it's around this time that the phrase, anything found to be true of E. coli must be true of elephants, was coined. And most of the formative work on the central dogma was done with the bacterium E. coli. And going forward, mostly E. coli and Bacillus, a handful of model organisms, were used for studies of the molecular biology of bacteria. So, and this work laid a firm foundation for work with multicellular organisms, even humans. And I remember around this time, when I was a graduate student, like in the mid-70s, lots of microbiology professors said, microbiology is dead, it's not going anywhere. You should move into studying a eukaryotic system Like you might have one biologist in a chemical engineering department or one in a geology department. And of course, there were the microbes that cause disease, but it's backwater. But fortunately, we didn't know what we didn't know, which is often the case in science. And the age of exploration of microbes was not over. In fact, it hadn't even started. And this is a map of the Atlantic Ocean as it was thought to exist in, 1947? And this is the map that Christopher Columbus had when he took off from Portugal to look for whatever there was on the other side of the Atlantic Ocean. And of course, Christopher Columbus and explorers who followed discovered the whole continent of North America. And that seems really important, but Carl Woese discovered a whole new kingdom that we didn't know existed. And this paper, The Phylogenetic Structure of the Prokaryotic Domain, published in 1977, was, a really important paper. Its publication and the New York Times announced the discovery of archaeobacteria as a new kingdom of life. And of course, the genius of the 16S ribosomal RNA molecule and of Carl Woese was that the 16S ribosomal RNA molecule is this really complicated, folded up in a complex way molecule that some parts of it can only evolve incredibly slowly, and some parts of it can evolve much more rapidly. So it's a molecule that you can use to look way back in time on the evolution of microbes, and then you can look at the evolution of microbes more recently. And so by 1990, this is what life looked like, with the branches of bacteria, archaea, and the eukaryotes. And this was thought to be the universal phylogenetic tree then, which was a huge radical change from the 60s, say. And now in 1918, Jill Banfield's group published this, 2018 published this view of life, which of course is made possible by huge advances in high throughput sequencing of DNA and RNA as well. And so here are the eukaryotes. I'm not dissing the eukaryotes, but really, this little tiny branch over here. And then the archaea are really diverse, and they're separated somewhat from all these bacteria, which are so diverse, and many of them haven't been cultivated yet. And there's still really a lot of discoveries to be made. So when I was director of the microbial course with Alfred Sporman, here's Alfred, in 2003, we had two instructors visiting. One was Jane Gibson and one was Ralph Wolf. And these were instructors when I took the course, when I was like 25 years old or something. And that's the thing, you get kind of attached to your instructors, and the instructors get attached to the students too. And Jane was here to help do the phototrophs, because she was an expert in phototrophs. And Ralph Wolf worked on methanogens, which we by then knew were archaea. And fortunately, he and Carl Woese were in the same department at the University of Illinois. Or Carl Woese might not have realized there were archaea for a good long time, because he just went around to every lab and got whatever bacterium they had and did the 16S sequencing of it. So, I'm pretty sure Ralph Wolf was here to supervise the Volta experiment. But Ralph Wolf had this, his father was a Quaker minister, so he was given to homilies, Ralph was. So he'd give these short, pithy lessons in life. And so he developed Wolff's laws of academic research. The first law being that unpublished data do not improve with age. The second law, if you're first on the scene, it's easy to make discoveries. The third law was the emotion generated in scientific discussions increases proportionately with the softness of the data being discussed. Yeah. So I know that every single microbial diversity student has engaged in the third law, because every single summer there's a discussion of how you decide what a bacterial species is. And it's like so fun, you know, to argue about. Fourth law, if you join a parade, this is sort of a corollary of the second law. If you join a parade, you become one of the marchers. Fifth, they who skate on thin ice must skate fast. Sixth, never stop in the middle of an experiment. That's like great advice for graduate students. And the last one is each brick in the wall of knowledge will withstand the weight of all future knowledge only if it is the full truth. And I mean, I really think about that every time, right before every time we send a paper in. And so I really took the second law to heart. If you're first on the scene, it's easy to make discoveries, because there were a lot of things that were interesting, but I wasn't sure if I could really compete with everybody out there. So I thought about this a lot, and I did a postdoc in a lab that worked on biodegradation of benzene rings or aromatic compounds. And at that time, there was a lot of interest in biodegradation of pollutants. And so one thing that we knew and that had come out was that oxygenase enzymes were really important for cleaving aromatic rings or benzene rings, and also for putting hydroxyl groups into the aromatic rings. And these were two things that had to happen in order for the ring resonance to be broken and the ring to be broken open. And these oxygenase enzymes require molecular oxygen. So they're very, very unusual enzymes, because molecular oxygen is a very difficult substrate to handle because it's so reactive. But when we were having lunch one day, because the lab always ate lunch together, it crossed my mind to think what might happen under anaerobic conditions. And Nick Ornstein, my advisor, said there was some work that had been done that compounds were degraded anaerobically, and there's people in this lab, in this room, who had already shown that. But not much work had been done on the biochemical pathways. So I knew some work had been done on a purple non-sulfur bacterium named Rhodopseudomonas palustris, but I had no idea, what to do. So I had to ask Jane Gibson. I did ask Jane Gibson, remember my instructor in the course, and she was at Cornell by then, and I was about to move to Cornell. So Jane rummaged around in her freezer, found our palustris strain that she'd isolated from Beebe Lake in Ithaca, and it grew anaerobically on benzoate. And we nicknamed it RPAL, and this is the color of RPAL, the same as Colleen's shirt that she's wearing. And it has this color because it's a phototroph, and the carotenoids and chlorophyll it makes are purple. And so, to make a long story short, I established my lab, and Jane and I worked together, and the people in the lab worked, and we elucidated anaerobic benzoate degradation, genes, enzymes, genes, and regulation. And the key was that you had to put a CoA, you had to convert the benzene ring to benzoate, which involves a carboxylation, which we did not work on. It's really a tough reaction, and others have. But then you need to have a coenzyme A put on the carboxyl group to loosen the ring resonance. And then there's this really powerful enzyme that reduces the ring. so it doesn't have any double bonds, and then it's easy to open up. So we worked on this over the years. And then because of this work, we had an opportunity to get the genome sequence of RPAL. And the genome sequence confirmed what we already kind of knew, which is that RPAL is the Swiss Army knife of bacteria. And it can do all these different things: photosynthesis, it can do aromatic degradation, carbon dioxide fixation, autotrophic growth with all kinds of electron donors, and it also does nitrogen fixation. And this, I want to give credit for this cartoon, which was drawn by Jake McKinley, who was a postdoc in my lab, but now he's a professor at Indiana University. So if we look at a blow-up of the Swiss Army knife, this is nitrogenase enzyme, and it converts nitrogen gas to ammonia and hydrogen. It has to make hydrogen along with ammonia. It's part of its catalytic mechanism. And this is like a really, really important enzyme on Earth, because it's the major way that we get nitrogen into the biosphere. We also have something called the Haber-Bosch process, which uses up 2% of the world's energy budget for making ammonia, but this is the main biological way to make it. And the nitrogenase compound enzyme is very, very complicated because it has to do a really difficult catalysis of reducing this triple bond in nitrogen gas. But when it does so, it requires a lot of ATP, very reduced electrons. And as I said, hydrogen is one of the products. Now, if you get a cell that's making nitrogenase and you don't let them have any nitrogen, but you don't let them have any nitrogen gas, then they make pure hydrogen. So for one thing, hydrogen is really easy to quantify. And for another thing, this was a time when biofuels were becoming really, really important to funding agency. So I thought it would be fun to see how much hydrogen we could get RPAL to make. And we had kind of a breakthrough because the nitrogenase enzyme, nitrogenase gene expression and enzyme synthesis are regulated at many different levels. But Federico Rey in the lab developed strains that synthesize nitrogenase and produce hydrogen constitutively. So he was giving a poster at a meeting, and he met Michael Flickinger, who is a professor at North Carolina State, who was experimenting with something called latex biological coatings. And so what Michael was doing was he was taking, he was working mostly with E. coli, and he took E. coli cells that weren't growing, and then he entrapped them in these sticky latex films. So the latex would be like latex paint that you go buy at the hardware store, but it doesn't have any fungicides or pigment in it. And so you can mix cells into the latex, and then they're not growing and they're entrapped. And so they can theoretically do 100% conversion of a substrate you give them to a product, because since they're not growing, they're not diverting any resources to growth. So Michael thought this might be good to try on RPAL, on hydrogen production. So this is Jimmy Gossi, who is Michael's grad student. And so Jimmy embedded RPAL in latex. And he made these really thin films that looked a lot like fruit roll-ups. Remember what those are? And then we cut the films into strips. And the strips were only 50 microns thick, but the density of bacteria was 10 to the 11th per mil. So the bacteria were just packed in there. And they were and the bacteria were expressing nitrogenase, but they weren't growing because we didn't give them any source of nitrogen. And so they just, when they're not growing, they just stay in the strips. We shine light on them and they made hydrogen, quite a lot of hydrogen. Here's a prototype that Jimmy made. You have to shine light on them. These are the bubbles of hydrogen that are coming up in the strip. And so, and we'd give the cells a source of electrons, which they need to make hydrogen, and they were getting the ATP that they need to make hydrogen from light. And we found that latex films of RPAL would produce hydrogen continuously for 4 months. And it's like, I mean, this is ridiculous. How can they be making hydrogen for 4 months? And so then a question came, how do RPALs stay alive and metabolically active for over 4 months when they're not growing? And so this really got me interested in bacterial longevity, because bacteria in nature, we know, I mean, there's so many bacteria everywhere we look. Colleen did a great job of describing that in quantitative terms, that if they grew at all, much at all, we'd be completely up to our ankles in bacteria in about a day, and then we'd be buried in them 4 hours later. So we know that bacteria on Earth don't grow very much, and that most of the time they're probably not growing at all. But this has been very hard to study in the lab, because non-sporulating species tend to grow to stationary phase and then they die. So you get, and this, I'm specifically talking about E. coli here. So you get a mix of live and dead cells, and they're all heterogeneous. It's really hard to study. So, but RPAL has a metabolism that supports longevity and growth arrest. And this is how it makes energy from light. It does something called cyclic photophosphorylation, and it has a reaction center proteins that hold chlorophyll and carotenoids. And light strikes the reaction center, energizes an electron that goes through a little electron transport chain, and then comes back to the reaction center. And that creates a proton gradient that is needed to make ATP. So that's how they make energy. But then they need to get carbon, and how they like their favorite carbon sources are organic compounds. So typically we give them acetate as a carbon source, and they use it to make more biomass, but they don't use any of the acetate for energy. They only use it for carbon. So this is what makes it so good to use RPAL as a model for longevity, because we can grow up the cells with a growth limiting amount of acetate, and then they'll stop growing when they run out of acetate. But if we keep light shining on them, they live for a really long time. Here's a 35-day experiment. And if we put them in the dark, they live for a while, but then they die. So one of the first things we found, and I'll just tell you a few things we found, is that growth-arrested RPAL continue to synthesize proteins. But they make fewer ribosomal proteins. So this is a proteomics experiment, and red means made more proteins at day 6 post-growth arrest and then at day 20 post-growth arrest. So they're making more of some proteins, less of others, and they're making fewer ribosomes, but that's like a well-known feature of cells when they go into stationary phase, that they down-regulate their ribosomes, but they still make as many translation factors as usual. So we identified longevity genes by TN-seq, and to be a longevity gene, to be a longevity mutant has reduced survival during growth arrest, but it grows normally. So that's our criteria for calling something a longevity gene. And when we got a list from TN-seq of genes that we thought might be involved in longevity, we picked out the genes that are conserved among all bacteria, because we wanted to learn something about how all bacteria stay alive. And 2 that we picked to look at are in E. coli called YBEY and ERA. And it's really interesting, because these are growing cells, and the red and orange traces are the traces for ribosomes made by these two mutants. And you can see they make pretty normal looking ribosomes compared to the wild type. But when these mutants go into growth arrest, they don't make any 30S ribosome, and they don't really make any 70S ribosome, and then they start dying. And what these mutants have is a defect in 17S ribosomal RNA processing, and growing cells have the same defect, but the defect isn't severe enough when they're growing to slow them down. The defect only becomes really critical when the cells are starved for carbon and in growth arrest. So we validated all these longevity genes, and we've picked ones that are conserved in other bacteria. And some of them, we have no idea what they do, so those are interesting. But for now, we're trying to track down the ones that are conserved, especially in other proteobacteria, because as Colleen said, and I forgot to mention, our pal is an alpha proteobacteria. So our pal conclusions, I think, is a good model bacterium to use in defining genes that are important for longevity in many bacteria. This is where my career has taken me. And some of the longevity genes are conserved and some have unknown functions. I've really been inspired by the microbial diversity course. I think to have the courage to just go and find something interesting and then study it. Now, but speaking of longevity, what about the microbial diversity course? I think we're good to go for another 50 years. The great thing about the course is the microbes are always the stars, not the instructors. It's the microbes that are the stars. And they're the foundation of the course is really to enrich and isolate new microbes. So the Van Niel legacy is intact, but each group of directors brings a new emphasis and flavor to the course and studies bacteria that you can isolate, studies them in situ in the environment using molecular techniques, first of 16S ribosomal RNA sequencing, and now we can sequence whole organisms that are in the soil that we can't isolate. So the Woese legacy is intact. But I think the big thing about the course is it's structured to empower students to make new discoveries. And so that to me is what makes this course so special. Now I just want to thank all these people who were microbial diversity course members from my lab. I know many of you probably remember Amy Schaefer, who still works in the lab. Yasu Oda's been with me for a long, long time and done a lot of important work in my lab. And Max Yin and Kieran Pector are the two postdocs who have done the work on longevity. And I want to thank you for thank the funding agencies for my funding, especially the Army Research Office. And I'm happy to take questions.
Ready to go? Thank you, Carrie. I'm curious, do my symbiotic mitochondria have longevity genes?
Well, I think of course, the mitochondria are derived from the alpha proteobacteria, of which our pal is one. And I think that the break point in aging, a big break point is the ribosomes and ribosome not staying intact and breaking down. And I think that's a key point, a key place that we're identifying in our longevity studies here too. Yep. Thanks for asking that. Yeah. Yeah.
I'm going to see if I can ask this in a way that makes sense. at the end, you were talking about the fact that macromolecules would have to be repaired
Yeah.
right, in a long living species.
Yeah.
So do we have any thoughts or data on like, just say like a transcription factor in a growing cell? Does the transcription factor over time just start to develop problems or does it, do you know what I mean?
In a growing cell. Well,
Right.
okay, so in a growing cell, you got to remember that the cells are dividing.
Right.
Even if the cells are only growing with a generation time of 6 hours. So they kind of make new ones every time they divide.
Right.
But when I'm not sure if this is what you're asking, but when they're not growing and they're going out for 35 days or 60 days, they're stuck with what they have.
And is there any data that being stuck with what you have is bad, right? Like, do those proteins eventually start to misfold or break down, or will they just go forever?
Well, we think they probably need to be repaired and turned over, some of them.
But do we
And
do
the
we know
ones
that?
that turn out to be tied to longevity genes are kind of the weak points. Yeah.
Do you know why some of the E. coli continued making at semi-normal levels the 50S ribosomes even when they were under growth arrest? I think I saw like the orange line. It stopped the 30S ribosome and the 70S ribosome growth.
Yeah.
Even when it was under growth arrest, it continued semi-normal levels of the 50S ribosome.
Well, when they're growing, they make pretty normal looking ribosomes. And they still have a processing defect. So why did they make normal ribosomes then and not when they're in growth arrest? That's an excellent question that I'd like to know the answer to. I mean, yeah.
Okay, we'll take one online and then one right here.
Do your bacteria genes have telomeres? Do they shorten with age if they do?
Oh, that's a really good question. Bacteria do not have telomeres, as far as I know. There might be some that have them, though. So no, they don't have them, and that's not an aspect of aging for bacteria, as far as we know. Yeah.
Hi, I'm a first-year PhD student, and I'm diving into studying longevity in a plant pathogen because it can persist in water for decades. And one of the things I'm looking at right now is adenosine metabolism and how that might contribute to longevity. Have you noticed anything with
Can you say that again? So what metabolism?
adenosine metabolism, specifically a AMP nucleosidase gene that I'm looking at right now? Have you noticed anything in your models?
Nucleosidases. I don't think any of those came up in our screens, but we're about to redo the screens in a way that I think will be more sensitive. So we'll be able to see. We do see DNA repair enzymes in our screen, for example. Yeah. Are we? Oh,
I'm coming, Colleen.
okay. You get a workout running these microscopes.
So Carrie, going from the lab back to the field,
Yeah.
do you have any sense if these are the same sort of mechanisms happening in bacteria in the wild? I had never thought about how fast bacteria multiply. I knew they could get to the moon in a day or
Yeah, right.
10 minutes, but I never thought about them around my ankles and then my waist. And,
Yeah. Well, we've just done that's a thing that we're starting to work on. We're starting to put the bacteria into our pal into soil, and it seems to survive pretty well under certain conditions. So what we have to do now is there's a lot, obviously, a ton going on in soil. So we have to first put them in sterile soil and then see how they do and see if the longevity genes are important in that situation, and then build up to more complex situations, but that's really important.
and can I just ask, is this a new field? I hear someone else is working on longevity in bacteria. I mean, this honestly, the concept of bacteria up to my neck is, I always thought it was viruses,
I think
protists.
we just wrote a review on it that's sort of summarizes what I think we know about all bacteria. And it's not new. It's been studied for a long time, especially by marine biologists. And, but they could never get very far. And part of that is they didn't have the tools at the time. A lot of it, I think, is they didn't have the tools at the time. Yeah.
So as a complete layperson,
Thanks.
I enjoyed thinking about this longevity of bacteria, and it kind of made me think about extinction events. And I'm curious if there's like a sort of mass extinction of bacteria maybe caused by humanity, and we should be thinking about conservation of these
Well, of course
things.
we should. That's a great question. Are there bacteria that have evolved on Earth that have become extinct? I mean, certainly some have, and it's because our atmosphere changed. I mean, cyanobacteria changed our whole atmosphere to oxygenate it, and the anaerobes had to all crawl into anaerobic environments. I'm sure some that had evolved then went extinct, and we're on our way to changing the atmosphere again drastically. So we'll see. Well, I won't see, but maybe you will. Yeah.
So we have time for just 2 more questions. I'm going to go with one here, and then I'm going to give the privilege to one of our trustees and Nobel laureates in the audience.
Thank you very much. The talk of growth-arrested bacteria and longevity genes make me think, as a physician, of the question of what role growth-arrested bacteria might play in the pathophysiology of infections and the treatment thereof. You know, we think of antibiotic resistance as, you know, ability to get strains that, you know, when they're, you know, growing, they just won't use this. But do these growth-arrested bacteria play a role potentially?
Well, yeah, so there's a phenomenon called antibiotic persistence. And what it is, which a lot of people have worked on, and what it is when you treat some infections with antibiotics, you'll kill off like 99.99% of the cells, but some will always persist and stay alive. And, you know, it's really hard to study because you can't rely on who you're going to have persisting. But I really do think those are cells that are in growth arrest for, you know, stochastically, you're going to get some that can stay in growth arrest for a long time and resist the antibiotics, because this happens with antibiotics that would only kill growing cells. Yeah. So it actually has analogies to. Oh, really? Yeah. Way off, but when you think about Yeah. 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 No, I think there's some parallels to be drawn with infectious disease. Yeah.
And our final question from Professor Avram Hershko.
Well, and there is some old literature that when bacteria are going into growth arrest, the rate of protein degradation increases, and it was thought that maybe this has a role in degrading proteins that are not needed to make a few proteins that are needed. So are these one of the
Yeah, that's one of the things. Yeah, that's one of the things we really want to look at. Like do a, you know, sophisticated pulse chase experiment to see which proteins disappear. Well, we know which ones disappear and how they're repurposed to go into other proteins. So.
Thank Dr. Harwood, and join us at SWOPE.
Thank you.